Hello, everyone. My name is Eric Lyons. I am the lead PI of Cybers and one of the PIs of the Agricultural Genome to Phenome Initiative, nominally in charge of these workshops. Just want to let people know that the Agricultural Genome to Phenome Initiative is funded by the USDA, and its purpose is to create a shared vision across crop and livestock communities. AG2PI holds a series of community building events, including field days, webinars, seed grants, and importantly, workshops such as the one that we're having today. Today's workshop is entitled The Structural Variant Detection in Animal Populations. This workshop is being held by Ryan Layer, who's an assistant professor in computer science at the University of Colorado Boulder. And two people in this group, Murad Chowdhury, who is a staff scientist in this lab, as well as Devin, who is a master's student in the group. Devin is going to be available for tech help on any of these workflows. In addition, we have Tyson Swetnam, who is a co-PI of Cybers and lead of Cybers' science team. He's here to help with any Cybers technical problems. We have created two breakout rooms, one specifically for the analysis help, which is under AG2PI help as a breakout room, and one for Cybers help. If at any point during this workshop you have any problems, please feel free to jump into one of those breakout rooms, and one of us will join in there to help you as best as we can. And with that, I'm happy to turn this over to Ryan and Murad. Thank you both again for teaching this workshop, and we're really looking forward to it.
All right, thanks for coming, everybody. So a little bit more setup here. I've put a link in the chat to a document. This is a Google Doc. This is a live, document, living document that we will update as necessary throughout this meeting. You can follow along with what we're doing. The intent is that you take this document with you when you're done. You share it with your labs to remind you of what we did and how to do it on your own. We'll eventually make this a PDF and post it somewhere, but we wanted to keep it going. If there's any questions or clarifications we can make on the fly.
Yeah.
Murad also wants, before I keep talking, he keeps asking me to, he wants a few minutes to tell you what you should download, so then I can kind of blab for a little bit while that stuff is transferring.
All right, so let me just, I'm going to briefly share my screen of this. So basically, before we get started, we're going to start off by cloning a GitHub repository, that I've basically created that has just a set of scripts that I will kind of be running throughout the process once I get to the workshop portion. So that Git repository is going to be located at github.com/mchowdh200. That's my username /sv_workshop. So basically, this is going to be located in, so when you log into the Cybers discovery environment, you'll log in and you'll want to launch a Cloud Shell, which will basically, once you launch a Cloud Shell, it'll bring you to just a simple like Linux command line environment. And from there, it will start off by putting you in a ~/work directory. And what you'll do is in that directory, you'll just git clone this Git repository. And once you've cloned that, it should take relatively no time at all. You just change directories into that workshop directory and then run a setup script. Essentially, all this script is doing is just creating a set of content environments that we'll be switching back and forth from. So yeah, basically while that's running, it should take like a few minutes, and then we can just have Ryan basically kind of speak as well. So basically after this has been run, we will then do download just some of the preliminary data that we'll be working on throughout this. And that's going to be done basically through the community data store through this. So the path for that will be basically downloaded through this command line environment. So we'll start off by running a command that's basically already pre-installed in the Cloud Shell called init. which basically, once you've run that, it'll prompt you for your Cyverse username and password, which will just initialize that so you can actually download from that data store. So once you've done that, then you can use a command called iget. And then basically you use this command, iget dash capital P, capital T. And basically then you can download this these two files here. Basically it's a BAM file for the two whole genome samples that we're using as well. All right. Are there any questions on this at all?
So we'll throw these commands into that document, so you don't have to try to get all of these things just right. So it's not there just yet, but we'll put them there shortly.
Yep. All right. So I'm going to go ahead and stop sharing now, and then I'll go ahead and put those in that document. All right.
All right. Thanks, Murad.
Yep.
So while people are scrambling to get those commands run, I just wanted to give a little bit of background. So our group is a computational group, and one of our areas of interest is structural variation detection. And we got into the non-human game a few years ago, looking at a few hundred salmon genomes. And in doing that, we found some interesting transposable elements in those genomes. That paper has some other attention in the fish world, and we basically replicated that same experiment with rainbow trout, and we're now moving into other species. To try to help mechanize that process, we are actually in the process of developing a kind of very, very easy to use pipeline that is the basis of today's workshop. So we're going to go step by step today, but in the near future, we will release software that can run the entire pipeline from beginning to end. The idea being you don't need any prerequisites. You need your sequenced files, your sequenced samples, and you need a reference genome. And from that, the machine will just or the code will take care of all of the intermediate steps. The purpose of this workshop is to dive into individual details, in case you have specific experimental design that needs to be adjusted from what the general workflow will be. Yeah, so we're very excited to kind of venture further into the non-human structural variant realm. Murad is going to run most of this. Murad and Devin have built this pipeline, so they're the best ones to talk about it. But please pipe up if you have any questions or want us to speed up or go slower. This is the first time we're publicly talking about this pipeline, so feedback would be super helpful, both in the steps and in the documentation. So are there any questions before Murad jumps in? So Devin's saying the commands are in the doc, so you should be able to copy and paste those. All right, great. Well, thanks for everyone for being here, and Murad, why don't you start?
All right, so let me go ahead and share once more. All right, so basically the way I'm going to do this is I'm going to basically be kind of bouncing back and forth between slides and then this Cloud Shell environment, which is just a command line like Linux environment. So basically the general outline of topics that we're going to talk about is just the process of calling SVs, as well as visualizing results and doing some sort of like visual validation, as well as like some automated validation of structural variants. So one part that we'll kind of be skipping but is present in the worksheet is the process of aligning sequence. We're skipping that mainly because running alignment on sequence is a very lengthy process, and running it even with like pared down data can take a very long time. So we're just going to leave that for the worksheet. Next step is essentially basically some pre-processing steps where we want to determine regions in the genome that might be problematic for calling SVs. So for example, regions that might have like abnormally high coverage or gaps in the reference genome that we're using. And then once we've determined the regions for that, we'll then move on to calling and genotyping SVs for population calling, essentially. So basically the main steps are calling SVs, merging the samples that we're calling with, and then finally genotyping and annotyping final results. And after we have a set of called SVs, it'll basically consist of us kind of examining some of these results, visualizing some of these SV calls with a tool that we've developed called Sandplot, which is pretty useful for visual validation as well as just looking at the regions that we've created. And then finally, we'll look into filtering out, possible false positive SV calls using either visual validation methods or using, for example, automated methods like using certain coverage metrics or using machine learning models for that purpose. All right, so next, so we've already kind of went through this download step. This would be if you wanted to just download it all at once. I don't recommend doing this because it will take a decent amount of time, at least in the live setting. That will take a decent amount of time, but it will contain basically all of the results of any commands that we're running today. So if you ever want to just see the entirety of what we've done here, it should actually be present in this public directory. All right. And of course, most of what we're going to do is also present in this SV workshop Git repository, which will have basically all the commands that we're using, all of the kind of scripts just put into discrete chunks for easy consumption. All right. So yeah, the first main thing we're going to start out with is this notion of filtering out problematic regions before calling SVs. And for anyone here, if it's possible, feel free to interrupt me if that's possible. I'm not sure what the privileges are with microphones are, but if anyone wants to interrupt me with a question, just feel free if I'm glossing over something perhaps that's maybe not obvious. All right. So many SV callers are plagued with false positives. Basically, in order to have a decent amount of sensitivity when calling structural variants, they will necessarily make a decent amount of false positive calls. And in short read sequencing, some regions in the genome are prone to low quality alignments of reads, and they can confound SV callers more easily than others. So, one common example is sequences that might align to highly repetitive regions. They might align to multiple different spots in a repetitive region, which can cause regions of abnormally high coverage, which will basically look something like this. So these images we have here are just some images using our tool Sandplot for basically visualizing genomic regions and some of the different signals that are associated with them. So for example, this gray region is essentially the depth of coverage of a certain genomic region. And these horizontal lines are basically visualizations of actual paired end reads for paired end sequencing. So you can kind of see some of the evidence that might support a structural variant. But in this particular case, for example, if you see like these two regions here are indicative of regions that have abnormally high coverage. So for example, in a region, so this particular genome that was the image was generated from was about a 60x coverage genome. So if you're seeing regions that are upwards of 200 or above, you're probably seeing some abnormally high spikes in coverage in what's generally a poorly behaved region for SV calling. And regions like these are going to cause false positives at a higher than normal rate. So even though an SV caller may have called this region as a deletion, it's unlikely that this was actually a true positive deletion, and it's likely going to be a false positive. Whereas this region here is a nicely well-behaved region where coverage is basically as much as you would expect at its highest point, and then at this point you see what is essentially like one copy loss deletion. All right.
I want to add a little context here. So this is a critical step, and especially in genomes that might be new and might have thousands of contigs and might not have the same attention as model organisms. So in the human genome, we have many annotations that we use to develop our exclude regions, microsatellites and other things that are known to be enriched for false positives. In many cases, non-model organism and non-human genomes do not have annotations like that. And they have many more gaps. And so this is a point of that we've spent a lot of time trying to figure out, and is critical for getting to a reasonable set of structural variants from which you can do things like population analysis or other standard variant analyses. So this is, I just wanted to kind of make a point that this is a very, very important step, and it is unique to genomes that might have many gaps or have many contigs or don't have the same resources associated with them as model organisms have.
All right, so moving on from that. So basically the first step that we're gonna essentially go for when we're trying to compile this list of problematic regions that we can use down the line in our SV calling is starting off with computing high coverage regions. So I'm going to kind of go through some of the tools that we use for that essentially. So we're going to start off with the tool for computing coverage across a genome. In this particular case, we're using a tool called most depth, which is going to be installed as part of that setup script that we have. But essentially what most depth is, it's just a simple command line tool that allows us to input any type of BAM or CRAM alignment file and just very quickly compute coverage across the entire genome. So in this particular example, I've just posted just an example command where you can call it, and then you can essentially select the window size by which you want to compute the mean coverage window by window. So in this particular case, for the demonstration, we're using a very large window, but maybe in general, you might not go for as high of a window, but it'll just run a little bit slower. But for demonstration purposes, we're just keeping this window large. But the idea here is that we're computing across each window just the depth of coverage across a particular sample in their genome. And the purpose of that is to basically then filter out regions that are having abnormally high coverage. So in this particular case, I'm going to go to our command line here. Oops. And what I've done is in that Git repository that we have, I've basically created a couple files. One is a script that will essentially run the most depth command, which is just the most depth.sh in that Git repository, which will essentially run this command. And I've also created a file called commands list. So within there, I basically just got a list of commands that we're going to be using throughout the demonstration. And I've basically started off by just commenting them all out. And then as we run through each one of these, I'll just uncomment them and then run them individually. So just for reference, I am basically running this or reading this using a command line text editor called Vim. For those that aren't familiar with that, it's not a very complex text editor, but we're just going to be using it just to do some very simple like commenting and uncommenting of code as we're running through this. So you don't have to be an expert in this tool, but we're going to just be using that. So we're going to start off with, at the top, I basically got the most depth command here, and we're going to be running this on basically the two genome samples that we had. In this particular case, we were running it on two Alaskan salmon whole genomes. We've basically reduced them to just one chromosome just for the demonstration, just to make sure that run times are reasonable as well as file times or file sizes. So we're going to start off by just uncommenting this. The way I do it is in Vim. If you're not familiar, you can just go over the cursor with your arrow keys and just hit the X key and it'll just delete characters. Perhaps some of you already know how to do this, but for those who have never used it, you can just use the X button or the X key to just delete individual characters and delete the comments. All right, so then by doing that, we'll go ahead and keep everything uncommented. And then in order to save this file, we will hit the colon key, W to save our file, and then colon Q to exit. I may have been going a little fast there. Does everyone understand that? If anyone is able to respond. If not, then we will basically go through and run that. So we'll basically just go by bash commands list, and it should just run that. So for reference, running most depth on these samples should take roughly 30 to 40 seconds for each sample. So it's going to be about a minute. And to verify that, I can use a command like top, for example, to see what's running in the background. And right now we see it's running. But like I said, I already have this run. It's running in the background, but I can examine the output of it just to kind of give an idea of what we're actually creating with this. So if I change directories into the most depth directory where everything is being output, I can, for example, look at the XH82 sample that we have and see just what's going on here. So xh82.regions.bed.gz. So essentially what we have is bed file format, chromosome start and end positions, as well as the computed depth of coverage across that window. So in this particular case, the window size was 10,000. So it's just going through in increments of 10,000 and creating, calculating this depth of coverage. All right. So, and it looks like that command is already completed. All right, so next, once we've computed all the depth of coverage for both of these samples, which we have now, we're going to go through and try to filter out any regions of high coverage using some sort of threshold. So in this particular case, I just pre-did a script for just calculating the statistics of it. A fairly simple script, but just to go through it, calling it get high coverage.py in the Git repository. There's not really that much to it. It uses the pandas library. So when you're in your cloud environment, you'll want to actually activate that conda environment, which we created. So we'll have the conda activate pandas. So now, basically the way that this works is all it really does is just loads the bed file into some sort of tabular format data frame. And then essentially we're going to use the depth column to basically compute the mean standard deviation of the coverage across a particular genome, and then just threshold it based off of whether it's significantly above the mean depth of coverage. So in this particular case, we just chose a depth of coverage that's 4 times above the standard, four times the standard deviation above the mean depth of coverage, and we'll just kind of determine that to be excessively high coverage, but depends on your application and the amount of coverage that your sample has been sequenced with.
And it depends on if you're interested in a particular element, it might occur many more times than 4. So if you have a gene that exists many times, you'll want to adjust that 4. That 4 is a nice general threshold for looking at genome-wide structural variations. But as Murad said, your experimental conditions might need for that to be raised.
Yeah. All right. So going back to the commands list shell in our shell file in our Cloud Shell, I'm going to basically just go through and I'm just going to recomment some of the code that we've already written since we don't want to rerun it. So in order to do that in the Vim text editor, if you want to start inserting characters, you actually have to press the I key. That's going to put you into insert mode. For those that aren't familiar with Vim, you basically press the I key and that allows you to type in characters. So for example, now that I'm in insert mode, I can now type in individual characters and still navigate with the arrow keys. And then if I want to exit the insert mode you will press the escape key, and that brings you back to just the regular movement mode. All right, so I recommented the old code that we're not running anymore, and I'm going to uncomment our get high coverage code, basically just running it twice, once for each sample, one for the XH82 most depth regions, and then one for the XH96 most depth regions. And then the second argument here is basically just what our output file is going to be named and where that's located. All right, so I'm going to go ahead and save that, and then I'm going to exit. So now we're going to run this commands list again, just with that code. And this should run fairly quickly because we're just going over a small file and just getting the regions with higher coverage. So let's kind of examine what that resulted in. So we saw the original bed file that just had the list of coverages there. So now we should have just regions with high coverage. So if I go back and we go to the directory that's titled high coverage or high cov, we go there. We should have two bed files, one for each sample. In this particular case, XH82, XH96. We'll just examine one of them just to see in general what it looks like. It's not going to look too much different from what we saw before, but in this case, it'll just be just the regions that had above 4x standard deviations above the mean coverage. Now, in this particular case, maybe 4x standard deviation is maybe a little bit too low of a threshold because 46x is maybe still slightly too low to be considered high coverage, but for the purposes of demonstration, that's fine. You might choose a higher threshold for your determination of what high coverage is. But the idea here is that we filtered out all the low coverage regions and we're left with just what we determined to be regions of abnormally high coverage. All right. Any questions on that? All right, so next, after we found the high coverage regions, another thing that we're going to look for is this notion of reference gaps in our reference genome. So in a reference genome FASTA record, anything that looks like a string of ends in the FASTA record is essentially a reference gap. And so the process of finding that is essentially just linearly scanning through your reference genome and just finding the start and end position of each one of those gaps. This process can be a bit lengthy depending on the number of contigs you have, as well as the length of each contig. So for this process, I essentially just pre-computed this. I do have the script available that created this in the gapregions.py in our Git repository. But for the purposes of demonstration, I'll just kind of direct you to the discovery environment data store where we have this located in this directory here, gaps, and then I will put this in our chat as the location for that. So I think that's, well, actually no, copy the path. So basically, let me do the chat. That's possible, or I might just paste it into this slide here. I can't seem to access the chat when I'm sharing. Or perhaps, let's see. Oh,
yeah, I can do it.
Well, I got it, but just in case. So basically what I'm doing is I'm pasting the path to the data store, and then you basically just use the iget command like you did prior, but with this particular path. All right, so now I've posted it there. Going back here. All right, so next, so we're not going to run that because that really just takes much too long in a live coding setting, essentially. But the next step is essentially taking both our regions of high coverage as well as our gap regions and just merging that into a single bed file. And if we ever have any overlapping regions or regions that are fairly close to one another, we'll also go ahead and merge those so we don't have too many small regions that we want to exclude, but we want to have just like a nice set of regions that are fairly uniform. All right, so this is basically just the process of concatenating most of these bed files and then using a bed tools merge. So I'll kind of go through each one of these steps kind of step by step. Some of this is just some basic like bash type commands, and then some of it is more like bioinformatics, like using bed tool stuff. So starting off, we're using the cat command, which is basically just concatenating various text files that we enter as our command line arguments. So first one is, for example, the gap regions bed file, which is basically just a 3-column file of chromosomes start and end positions of wherever the reference gaps were. The next part is the high coverage bed files, but one wrinkle to this is that these bed files will have 4 columns as opposed to 3. So we do a little bit of extra processing to this to basically just cut out only the first 3 columns. So that's essentially what this command is doing. We're taking the original bed file, which had 4 columns, and then we're doing a cut command, which will essentially take, in this particular case, the first 3 columns of that bed file, and then it'll output that as the file, as opposed to using the base file. So we're doing that for both the XH82 sample as well as the XH96 sample, and we're just concatenating all of those into a single bed file. Next
this would
part
be a
is,
point to,
yep,
hang on, Murad.
go ahead.
This, if you had your own annotations about regions that you knew were problematic in your genome, this would be a point to where you could just include them in this list. So here we have 3. Obviously, if you have more samples, you would have more of these cut commands, but you could also imagine having some other lists of, centromeres, or those are probably gap regions, but you could have other regions that are problematic you could include in this list as well.
Yep. All right. So basically we start off by just concatenating all these things into one file. And then we use a Unix pipe to basically just continue that into a bed tools command. So we start off by just sorting them all together, and then we use bed tools merge, which is essentially taking a bed file and then taking any regions that are within 1,000 base pairs of one another in this particular case, and merging them into a single region. So if you have 2 regions, they're either overlapping or within 1,000 base pairs of one another, it'll merge them into a single region. So it just creates a more uniform bed file for us to use as our exclude regions. All right, and basically that will also be present in our commands list. So let me go down there. In this particular case, so let's go ahead and just recomment our old commands that we have just run. And so next is, so we're skipping the reference gaps, and then next we are going to do the merging step here, which is right here. So just uncommenting these. All right, save and quit there, and then we just run that real quick. So run that. That's going to be located now in our exclude directory. So I'm going to go back to our data directory. That's where our exclude directory is. So cd exclude, and now we can just kind of examine what that is. So we will just kind of look at that. It's just a combination of all of the regions that we've gotten from the reference gaps, and then the high coverage regions from both samples. So in this particular case, it resulted in roughly 4,000 regions that we can now use. In this particular case, it's just the first chromosome, since we just pared down all of our data to just one chromosome. But in reality, it'll probably be a lot more than this, depending on how you determine high coverage and if you're using the whole genome. All right, so now we have our exclude file. So this is essentially mostly just like pre-processing steps that we're going to go through before actually calling structural variants. So moving on to the next step, where we're actually getting into the process of calling SVs. There are multiple different types of SV callers out there. The one that we typically use, just due to its kind of ease of use and ability to kind of string together various steps in the process, is this tool called Smooth, which basically combines both an SV caller, Lumpy, and then a genotyper which is SV typer, and basically allows for fairly easy process of calling SVs for population calling. So basically, in the particular case of what we have in our setup script, I've created a conda environment for smooth. So we just conda activate smooth when we want to run that, and that will basically have that stuff installed there and any dependency that's required for that. Basically, the process that you go through is you want to call SVs in each sample individually, and then after doing that, you'll go through and merge samples into a single Bcf, and then do genotyping at the end, basically. So starting off, we're just going to do the SV calling portion. So for this, I'll just kind of go through the actual script that I have for that. So in what I've called call svs.sh in the Git repository, but it's essentially just a single call to smooth, and I'll just kind of go through the parameters that we use when we actually call svs using this method. So first off, we want to run smooth call. The name parameter is essentially just the prefix for the results Bcf. So like what I typically do when I'm calling on single samples is this name will essentially just be whatever your sample name is, for simplicity. The FASTA is going to be just the reference genome that you include, which was present in the data store for those that missed it. Let me go back there. So our reference genome is located in the data store, and it's basically located at this location here. So I'm going to copy that and put that in the chat just in case you missed it. Copy path. What's happening here? Oh, there we go. Oh, now the chat's not popping up. So maybe maybe Devin can copy and paste both this and the FASTA index into the chat for me if you're able to do that. But basically, that's the reference genome that we would include there. The exclude flag is essentially going to be the path to the exclude bed file that we just generated. So we can use that to basically prevent the SV caller from considering those regions to begin with. So there's kind of two processes, or two ways of excluding regions when you're calling SVs. One is to just do this before you even call SVs, is to use this like exclude file, or you could call SVs on the whole genome and then remove SVs that intersect with the exclude regions. Now, typically, you'd want to just do this prior to calling SVs because it'll just reduce the amount of time it takes to actually call SVs, because if you're doing it in a lot of these regions, there will just be a lot of discordant reads that will essentially confound the SV caller and cause it to take a lot longer to actually complete. So typically we want to just use the exclude file as we're calling SVs. And then next, the output directory, fairly self-explanatory. That's just the path that you want to output your results to. In the case of smooth SV caller, the number of processes has a set of diminishing returns. If you go above 4 processes, it tends to not really have much of an effect. When you're doing population calling, it's better to just set processes to 1 and then just parallelize over individual samples instead of going for multi-processing for the individual sample. The next thing we do is we actually do genotype individual samples, but the main purpose of that is really just to remove any obvious non-SVs, anything that's just the homozygous reference genotype, so that'll just kind of reduce the amount of data that we have later on when we're combining data together. And then this particular flag, this no extra filters, I basically just included this for the demonstration purposes because it does take a decent amount of time to call SVs, even when you're restricting your alignment file to just a single chromosome. In testing, it took almost an hour to call SVs, even on a single chromosome. So when basically, when you use the no extra filters flag, it basically just makes it a little more permissive with the types of SVs that it's calling, essentially. So you'll, in this particular case, we'll end up with potentially more false positives, but this is really just for demonstration purposes. And then the final parameter is just the alignment file that we're going to use. All right, so next is just kind of seeing that command in context for our shell in our commands list. Now, in this particular case, I've also pre-computed this. It should be able to run here, although even with the no extra filters flag, it'll take roughly maybe 10 to 15 minutes for each sample to run. So that's still slightly too slow for like a live coding context. So I do also have that located in the data store as well. So first, I'm just going to go through and just recomment some of this existing code. And then basically over here, we're going to uncomment this. I'll just uncomment one, but I'm not going to actually run this at the moment because it will take a little bit too long in the context of what we're doing. But essentially, we're just running this once for each sample. All right. And the location of that is going to be basically in the same data store area that we have here. And for each sample, we have an XH82 directory and XH96 directory, which will basically have all of the SV calling results located in a VCF in there. And I will basically examine that in this next step here. So since this takes too long to run, I'm just going to pretend that we did run it. But now, so basically, once you've downloaded that into the data directory, it's going to be located in one of two directories, one for each sample. So I'm going to just start off by looking at the XH82 sample. All right, so basically when you run Lumpy and SV Typer, it basically outputs various bits and pieces. For example, like some of the evidence that it extracted during the SV calling process will be located here. But what we're interested in is essentially just the genotyped VCF of that particular sample. So in this case, for the XH82 sample, that's going to be the XH82-smooth.genotyped VCF. While we can view this VCF using standard text editors, its VCF format, especially after calling SVs on a single sample, is still not very human readable, as you can kind of see. Essentially, a VCF is a tab separated file by region. So each line is a region that was called that contains information like the chromosome start position, end position, but also various other annotations that are left by the SV caller. And in general, the VCF format isn't really meant to be human readable, but rather something that you would query using a tool like Bcf Tools to then output into a format that's a little bit more human readable, which we'll be doing a little bit later on. But you can still examine various things here if you know what to look for. But in this particular case, we will not examine this that much further since it's not really meant to be read as such. All right. So basically, this is post calling SVs. Now we have two individual sample VCFs of SV calls. So the next step is going to be essentially just merging both of these VCFs into a single VCF. So you can't just take the regions of one and the regions of the other and just put them into one VCF normally, because one sample and the other sample may have two different sets of regions and different numbers of regions. So we have to basically do a specific merge step that basically takes regions that weren't present in one and regions that weren't present in the other and just basically make them have the same amount of regions. But that can be a fairly complicated process, but luckily Smooth does have a utility that allows us to just do this without really having to think about it too much, called Smooth Merge. Essentially, all it does is you just provide it with the prefix name of what you want to call it. In this particular case, for example, I can call it merged. You provide it with your reference genome in your output directory, and then a list of VCFs that you want to provide it with to merge. So
the other critical step in this merge process is to identify structural variants that are common among the samples. And this is a little bit of a tricky process because in many cases, structural variants are not, especially in short read data, are not identified down to single base resolution. There's going to be a confidence interval around the breakpoint. So you might know the breakpoint is between two sites plus or minus 10 base pairs. And so what this merge is aware of that ambiguity between calls and merges things as long as they are reasonably the same structural variant. So that's, as Murad said, is a very complex process. But the whole point of this step is to come up with a single list of structural variants that were observed in at least one sample. So it's kind of your master, we often call it the sites Bcf. It's like your master list of structural variants where every structural variant will exist in this list exactly one time. So this is kind of our merge step. We call variants in every sample individually. We take all of those samples, we do that in parallel typically, and then we merge them all to a single master list of variants, from which, as Murad will talk about, we then get the genotypes of every sample for that master list.
All right. So after merging, so that's basically where we actually go through and then start to genotype variants and annotate regions based off of various metrics. So, like we were saying before, even after merging, it's safe to assume that SV regions that are present in one sample but not the other probably implies a homozygous reference genotype. But tools like SVTyper and duphold, which is also part of the genotyping process, they basically annotate all the regions of the VCF, including regions that didn't necessarily have a variant in it, and it will annotate those regions with various coverage metrics, as well as various like number of alternate reads, number of reference reads in a particular region. So it's good to actually genotype every single region, even if you know that it wasn't a variant to begin with, for statistical purposes. So basically we'll be running this genotyper on all regions for all samples and basically get the annotations for that as part of the final VCF. So
we should note one, sorry, Murad, for keep on interrupting you.
That's okay.
The first step of calling structural variants, so you see up here like the XH82 joint smooth, or not that one actually. So yeah, the individual calls, using those genotypes are not going to be as reliable as this next step that we genotype after merging. So you should definitely treat these individual sample files as intermediate files only, and base your analysis. We call it a squared off VCF, where you have the master list of calls and you have a genotype for every sample for every call. And that makes all population or any other sort of association analysis much more straightforward than trying to look at individual samples.
All right. So yeah, so basically right now I have some extra output files that wouldn't have been present yet. For example, like this joint call, that's something that's going to happen afterwards. But currently we're at the stage where we have just this initially SV called genotyped sample, where it's just a single sample VCF. And the next step was then we merge those into a single VCF, which we're then going to regenotype every region to get all the annotations and everything in there. And that's essentially what this smooth genotype invocation is going to do. So basically, the smooth genotype, very similar to doing like a smooth call, but just a little simpler in terms of computation time. So that's going to be in our regenotype shell scripts. But essentially, we're doing some additional annotations. So for example, we're running a tool called duphold, which is essentially a very useful tool for annotating duplications and deletion events using various coverage-based metrics. So for example, duphold can basically measure like the fold change of the between the breakpoints and then in flanking regions, and determine like a number of fold change between the, if a region had a certain amount of coverage here and had a certain dip in coverage, then the fold change will be basically like less than 1, for example. And you can use these types of metrics as kind of sanity checks and as like post-processing filters to determine whether, you know, certain deletions or duplications are in fact true positive regions or false positive regions. So it's a very useful thing to run on your genotyped variants. This remove PR statement is basically, like we were mentioning earlier during the merge process, every SV breakpoint that's called by the lumpy SV caller has probability distributions around each of those breakpoints, and that's used as part of the merging process. But those probability vectors are very unsightly when you're looking at the raw VCF. So removing those after you're done with that process is usually a good idea. So if you ever want to actually read the VCF, you won't be inundated with long strings of floating point numbers. So that's the main reason that's there. In terms of multi-processing potential, actually, the genotyping is a lot more easily parallelized than the SV calling itself. So you can actually get linear increase in speed by increasing the number of processes as opposed to parallelizing by sample when you're doing just the first SV calling step. The rest of the stuff is fairly self-explanatory. For example, the reference genome is the FASTA. Bcf is the merged Bcf that we just created. The name, again, is just the prefix for the file that we're going to create as an output. So in this case, I just said, you know, name dash joint. So for example, sample name and then joint, just to distinguish it from our prior SV VCF that we created. So in this case, our initial one was, you know, xh82-smooth.genotyped, whereas the regenotyped one that we created is going to be xh82-joint-smooth.genotyped. So just as a distinction there, out directory, and then the output band file. So very similar to the call, smooth call command that we made, but just specific for genotyping our merged VCF. All right. And so after we regenotype, then we will basically have, in this particular case, since we have 2 samples, we'll have 2 separate regenotyped VCFs. And in this case, now each of these VCFs will have the same number of regions, which have all been annotated. So they'll all have genotypes at this point. So now it's safe to just do a simple merge operation as opposed to a more complex merge operation. So in this particular case, that's what you would usually call a paste operation, essentially. So we have this smooth paste operation, which essentially just takes a list of VCFs. But in this particular case, the condition is they have to contain the same number of regions. They have to be the same regions in order for this part to work. So this is essentially the final merging step after you've already genotyped your variants. And so the command is very similar to the smooth merge step, where you just provide it with your prefix name, your output directory, and the list of VCFs. And so in this particular case, I just made a name of final just so we can make a distinction between our merged VCF. So
hang on, Murad. I just want to make it clear that, so we did 2 types of merging. We first took all of our individual sample variant calls, and we retained variants that were observed just in one sample by themselves, and we combined variants that were observed in multiple samples to come up with one unique list of structural variants. That was our first merge. This merge is simply taking the genotypes for every sample for all of those, for that master list of structural variants. And you can kind of think of it as a matrix where you're just adding columns for every column. So in this case, we'd have 2 columns, one for XH82 and one for XH96. You might have hundreds or thousands of samples here where you're just going to be tacking on extra columns to that matrix of genotypes.
Yep. All right, so let's just briefly examine what the joint or the final VCF would look like, essentially. So in this particular case, I placed that just in the regular data directory. So that's going to be in the final.smooth.square VCF. So let's look at that. It's going to be a little bit easier on the eyes, but still not very human readable just because the lines are extremely long. It's really just meant to be queried But let's see. Format. So one thing to notice is just this kind of line here, the header that will kind of just show what each column represents. So you start off with your chromosome start position, some various information using like the ID of the variant, what the reference allele and the alternate allele is. But the things that we're usually interested in when we're querying a VCF is something like the information tags, which are basically semicolon separated bits of information that are specific to the region. So for example, like the SV type, in this case, it's a deletion. It'll tell us what the length of an SV is. In the case of deletions, they report SV length as a negative number. It'll have information like where the end position is, strand information, stuff like that. But then after that, you have what's called the format tags, basically, which is essentially the sample specific information. So that would contain stuff like the genotype, various annotations, for example, like duple annotations or SV typer annotations. And you'll have one column of these formats for each sample. So for example, in this particular case, we had two samples, XH82 and XH96. So we're going to just have two separate columns of format tags in this case. Now, of course, it's very difficult to read these things just from the VCF itself. So quite often what we like to do is use some sort of query command to basically just extract what we're looking for, whether it be a particular sample, or a particular SV type, and then we can also kind of format our queries to be a little bit more human readable, for example, in something like a bed file format. So what we do for that is use a tool called Bcf Tools query.
Hang on real quick,
Yeah.
Murad. So I think it's worth, for people who have not looked at structural variant VCFs, but have looked at SNP VCFs, it's worth making a couple of things clear. So one is this reference, I picked the worst possible color. This reference field and the alternate field. For SNPs, you might expect there to be like an A here, and then the alternate allele being like a G or something. The standard for structural variation for VCFs is a little bit squishy, and you might observe different things here. We, for the sake of making the lines shorter, put an N for every reference. And then for the alternate, we just put the structural variation type. It could be a deletion. Right here, you see a duplication. There's inversions, insertions, and the general category is called BND for break end. Other tools might choose to actually give an allele here and give the base pairs deleted here, which, as you might imagine, for let's say a megabase deletion, you might end up with 1,000 characters in this, or 1,000,000 characters in this field. So we've chosen for a more compact representation. So the fact that there's N there is not to be worried about. And then this just tells you the type. That type is repeated out here in SV type. This SV type is a much more standard. So for tools that would actually put the sequence in the alternate field, they will then identify the type of the variant in the info field. So SV type del or SV type dupe, those are very standard annotations for structural variants. Since for the way that we represent it, SV-len is going to tell you how long that duplication or deletion is, and then the end will tell you so pos right here will tell you where the variant started, the first base pair of the variant, and then end will tell you where it ended. So if you took end minus start, you'll get to the length. The other interesting bits here is the imprecise tag. So I talked a little bit before about we often do not have single base resolution on our structural variants. And this is saying this one in particular, we do not have single base resolution. And what is that resolution? Well, it's pretty bad in this case. So CI pos is a tag, and this one is broken over 2 lines here. So the start of this is at 1755.89. That's plus or minus 578 base pairs downstream and plus 29 base pairs upstream. So we've localized the variant to exist somewhere in that, roughly 600 base pair region. We think the most likely spot is at 1755.89, but it's somewhere in there. And then you have a similar case for, we think the most likely end of it is at 5961.04, but that's plus or minus here, CI end, 30 on one side and 533 on the other side. So I'm not going to go through any more, but those are just, if you're trying, oh, you think you moved the line. My circles are no longer lining up, at least on my end. I'll just clear that. All right, so just to kind of give a little bit of a tour of a structural variant VCF, they do look quite a bit different than a SNP or indel VCF. I actually think right now, so we were at exactly an hour, we're halfway through, and I think we're making good time. So I think it would be worthwhile for taking maybe just a 5-minute break where we can see if anybody wants to catch up. Devin, Murad, and I are happy to answer questions either over text in breakout rooms or in this channel, or if anybody else has any other questions. I know Tyson can handle some Cybers questions, but let's just. So I've got 103 on my time right now, so we'll pick back up. Murad will pick back up on his steps at 108, and we'll just hang out here answering questions, so everyone can kind of get caught up. I know we're kind of trudging through this a lot. Ideally, the worksheet, which I will paste the link to that back again. Give me 1 second. We're following this. All the steps that Murad has are in this Google Doc that we are updating live. So if there's errors, please let us know. But yeah, so let's just hang out for 5 minutes and then let people go to the bathroom, catch up, or ask questions.
Yeah, definitely ask me questions, because I feel like sometimes I might gloss over something that is obvious to me but not obvious to others. And if something is even slightly non-obvious, just feel free to ask.
There was a question earlier about installing all of this locally. Or hold on, let me wait 1 minute to see if there's a question before I start talking.
Okay. Yeah, I think I glossed over that a bit.
All right, so there was a question about what all needs to be installed if you're not doing it in Cybers. And so we specifically chose this environment called Conda, which it's a little bit complicated and doesn't always work, but it's the best way. You can kind of think of it as it's a virtual environment. So it's not a full virtual machine like you might have heard of as running on the cloud. Tyson, do you see someone jumped into the Cybers troubleshooting breakout room? So it's a virtual environment. So it will, once you start it, you can start installing all these tools and specifically customizing your runtime environment. And those tools and their specific versions and all those settings will be isolated to that virtual environment. So you can have multiple, and we do have multiple different virtual environments for different processes. And so these commands here that Murad has are assuming that you have Anaconda or Miniconda installed, assuming you got that far. And even if you're on like a server or a machine that you don't have root access to, you'll still be able to follow these steps, except for the very first one, the bed tools one, you'll need root access to. But all the following steps you can install locally, just in your environment without administrative access. So that is in the setupbnb.sh to follow.
Yeah, and one thing I kind of want to add is I probably should have went over this setup environment script a little bit in more detail, but I think I just kind of glossed over it. But essentially, the way I kind of devise this scheme is that anyone with a Linux environment should be able to run this in roughly the same way, whether you have like a local Ubuntu machine, or if you're working in for example, like your university's HPC Linux environment, should be very similar. Like Ryan mentioned, the one thing you might not be able to do is use sudo, which is something that an administrator would be able to do, but you can easily also install bed tools using something like Anaconda as well. Essentially, normally when you're doing individual steps, you basically create these virtual environments so that you can just have like very small siloed off environments so that your dependencies that you're using aren't interacting with one another. So in the context of like this demonstration, having a million small environments can be a little bit cumbersome, at least for demonstration purposes. But oftentimes what you do is you will have some sort of workflow management tool. For example, like something that's very commonly used in bioinformatics is Nextflow or Snakemake. These are basically these workflow definition languages that essentially can manage individual content environments for various steps of a data processing pipeline. And basically by having your environments be as narrow as possible, it basically ensures that all your different steps aren't necessarily like interacting with one another in adverse ways, which is very common. So, one example I have here is Smooth is a tool that actually relies upon Python 2. But of course, Python 2 has long been kind of deprecated by a lot of library makers. So oftentimes, a lot of modern libraries will use Python 3. So if you have an environment that relies on Python 2, it can cause some adverse interactions with some other libraries. So having these two things in separate environments is important. And essentially, for the demonstration, we've just created very small environments for most of the individual steps. For example, the most depth step will have its own individual conda environment. We had an individual environment for running, for example, pandas. And then same thing for like if we need to use BioPython, we'll just have that in a single environment. But essentially, in this setup environment, we've kind of annotated each individual environment with a little bit of an explanation as to what it is and what it's used for. All right. Are there any other questions regarding this stuff?
I think it's because you're doing such a good job. No question.
Oh, I hope so. Either that or switched off. But so how long has it been? We got any time left? Or
I think we've got 50 minutes, so I think we can
okay.
Go ahead and get started again.
All right, so let's get back to the process of querying an SV from a VCF or a set of SVs, because like we were mentioning before, even if you know what to look for in a VCF, this is still a very cumbersome process for just looking at, okay, this is SV type del, this is, CI pos minus 30. It's very difficult to look at this stuff, as opposed to like looking at something a little more sparse and compact, like a bed file format, which is really simple, just has, chromosome start, end point, and then maybe just some extra tab separated annotations that you're interested in. So one common thing that a lot of people like to do is use something called Bcf Tools to basically query certain types of SVs from a VCF and then put them in a certain type of format. So let me kind of just very briefly kind of demonstrate this. So in our Smooth environment, we should already have Bcf Tools installed. But essentially, if you want to take a VCF and query it for a certain sample, you can use -s and then the name of the sample you're interested in to just get that particular sample. You can use -i as basically an include flag for any type of format or info field, and then use various logical operators to basically use that to include certain things. So for example, I have a -i SV type equals del. And so that's basically just going to include only SVs that were deletions in this particular case. So if I do -i single quote SV type equals, and then you have to do double quotes around this, del, then that will basically just query all the deletions inside that file. But of course, we want to also format it, so we can use the -f flag to basically make the format of this into anything that we want. So one common thing that I like to do is basically take a VCF and make it look like a bed file format. So fairly easy thing to do. Basically, if you don't want to get the chromosome, you use % all caps chrome, and then put a /t to do a tab separation. The start position is called pos /t, and then the end position is an info tag. So you basically have to do %info / and then end to denote that this is an info field. So we'll say %info/end. And then anytime I need to look at something in the format fields, which is a sample specific field, you have to actually enclose those things in square brackets when you're doing a Bcf Tools query. So for example, in this case, I wanted to query what the genotype was of the particular sample that we had. So we had to do square brackets %GT for genotype. But you could use this with any other format field as well. So /t square brackets %GT. And if you have multiple samples in this query, this thing is going to be repeated once for each sample. So anything that's within here will be repeated, n times, where n is the number of samples. All right, so, and then of course you want to terminate this with a new line character, so that's a \n. So if I do that, and then nothing else, so of course we have to provide it with the actual VCF file, so final. So if I just do this, and then I'll just get the first few ones with the head command, this will essentially just give us a simple bed file format of just the information that we're looking for. So if I only wanted to see what chromosome start and end and the genotype was for the given sample that I was looking at, this is a much easier to read type of information as opposed to looking at the raw VCF. Even if I'm just looking at certain lines, it still gets very cumbersome. So doing this is much easier. We can easily change this query to be any other format field we want. So for example, another format field that I like to look at is something called the DHFFC, which is a dupole annotation, which basically gives me statistics on what the fold change is. So for example, if you see a fold change of 1 between the region and the flanking regions, this will basically mean that it's kind of like roughly the same coverage. If you see something that's less than that, that just means that there's a dip in coverage in that region compared to its flanking regions. So for example, if you see a 0, that means that essentially the average coverage within the breakpoints of this deletion was 0. So that's perhaps the reason why this was called as a deletion, for example. All right. Some additional things that we did in this particular command here was I did do an additional pipe to a grep command, which is essentially, if you're not familiar with it, grep -v will essentially match any type of pattern that I provide it with and then exclude that pattern. So for example, if I was only concerned with getting just homozygous alternate or heterozygous deletions, then maybe I want to exclude any genotypes that had a homozygous reference. Or if you see a dot/dot in your genotype, that means that the genotyper wasn't able to make a genotype. So I also want to go ahead and exclude those as well. All right. So then, doing some command like this will basically give us a set of deletions that were either heterozygous or homozygous alternate. So in that particular case, do I have that here? Don't, but I can basically just run that command, which I do have present in the commands list. Oh, I already had it. All right. So before I do that, I'm just going to go ahead and comment out my existing stuff here. Oops. All right, so the query is located here. Actually, I can get line numbers from this as well. All right, so it's present on line 86. So basically, I'm going to uncomment this and then run it. Basically, it just has the same commands that I have on the slide. Save and then quit. So I want to run the commands list. And so now that should be present in our data directory. Let's see, where is that? Let's see it. Oh no, I see it actually. It's right here, xh82.del.bed. So now we can take a look at this. sh82.del.dead. So like before, it's basically just the regions, but keeping only the genotypes where there was some sort of variant detected. All right. So now we can easily query regions, but that's not the the be all end all. We can also print various statistics and metrics for the validation of these types of variants, because one thing that's still common even after providing it with a set of good exclude regions is that most often you will see SV callers they'll be making a high number of false positive calls just by nature of the fact that in order to maximize the sensitivity of a SV caller, there's just going to be a fair number of false positives even after our initial filtering steps. So one thing that's commonly done is using various metrics for filtering these SVs, or by using some sort of visual validation method by, for example, looking at the coverage profiles, looking at the paired end reads and split reads and seeing do they match up with your expectation of what an SV looks like. So for that purpose, our lab developed a tool called Sandplot, which is essentially a command line tool for SV visualization. But in general, you could use this for just visualizing any type of genomic region, but it's tailored mostly towards structural variation in this case. So the idea is that given any BAM or CRAM alignment file and a set of regions, for example, in a BCF or a VED file, then we can just quickly generate images of these regions and be able to verify whether these SVs are true positives, false positives, or look at any other various properties of interest through this. So for example, in this example of images here, we mentioned earlier, but basically this gray plot is essentially the coverage, the depth of coverage of a particular sample at a particular genomic region, which is basically the x-axis coordinates here. So in this particular case, this is a human genome sample that we took this image from, but it's basically on their chromosome 4 from this point to this point. And the SV that was called in this particular case was some sort of deletion with the breakpoint spanning this region here, which we can kind of see just from the dip in depth of coverage from these 3 samples. So for example, this plot is essentially what looks like a heterozygous deletion or 1 copy loss. This looks like a homozygous alternate deletion, which is essentially like a 2 copy loss. And same thing with this is another heterozygous deletion. But additionally, besides just depth of coverage, in paired end sequencing, you can actually visualize the paired end reads. You can also visualize split reads. Basically using in Sandplot, they will be displayed as these horizontal lines that will essentially span the points at which they were aligned to in the reference genome. So we have 2 separate y-axes that essentially show 2 different types of metrics. On the right side, y-axis, we show the depth of coverage. So in this particular case, this is like 60x coverage, and then you see the dip here. But on the left side, you'll see what the insert size is. And so basically, when you see horizontal lines that are aligning with this point in the y-axis, that means that the insert size of these particular paired end reads was 3,000, for example. Now, SV callers for short read sequencing essentially use information like the length of insert size of certain reads, as well as the presence of split reads to basically use that as evidence of structural variation. So typically the mean insert size will be some normally distributed number, for example, like 500 in a lot of cases. And what will happen is if you see something that was much longer than that by a certain amount, so for example, in this case, a lot of these paired end reads seem to be above 3,000 insert size, where one end is aligning here and another end is aligning here. That's essentially like evidence that the SV is essentially spanning these 2 regions. In this case, that there was like some sort of deletion going on here, because in the reference, you would normally see these 2 regions next to each other, whereas in the case of like something deleted, it'll be much farther apart. So this is basically another way to essentially visually validate that this is a correct SV call. But we can also use this as essentially let's see if I can find a good image of this. I had previous. So we had some Sandplot images here where, this is a pretty clear true positive for a heterozygous deletion, but what happens if I call an SV and I generate a image of that region and it looks something like this? This was called as a deletion, but in my eyes, it doesn't look like that at all. It looks like just like a region with poor quality alignments where there's lots of high spikes in coverage. There's some regions where there's just nothing aligned, but the overall coverage seems to be low in this region anyway. So it seems like there was just a lot of bad quality alignments that are basically confounding an SV caller. But to the trained eye, you can essentially look at a region like this and say, well, this is clearly a false positive. So you can essentially use this tool as a way to visually validate your set of SV calls. All right, so moving back to where we were. So basically, in our environment, we created a conda environment for Sandplot as well. So if we conda activate Sandplot, you can basically either call the Sandplot plot command just for single regions, or you can incorporate this into other scripts where you're basically doing this, genome wide or across an entire set of samples and plotting the regions from a Bcf, for example. But in the case of just calling it on a single region, Sandplot plot, it essentially just takes various parameters here. Essentially, the main ones to consider are essentially just the genomic region. So for example, the chromosome start and end position, the SV type that you're looking at, that's just for labeling the axes of the plot. But then you provide it with a alignment file, which is like a BAM or a CRAM, and then it'll output just a simple PNG file. And so we, in our Git repository, we created
you can include, it can do multi-sample visualization. So for as many BAMs as you include, just with spaces, it'll add another subplot. So you can, do, if you want hundreds, 10 is about the most that you can look at, but it can do multi-sample visualization.
Yep. All right. So in our Sandplot plot script, essentially we're doing a little bit of pre-processing here. So in this particular case, we're looking at just a single invocation of Sandplot plot. But of course, our SV Bcf had a lot of regions in it. So let's actually take a look at how many regions our bed file actually had in it. So we, for example, if I want to look at all the deletions as Sandplot images, we can take a look at how many lines does our bed file have. So that's xh82.del.bed. I'm using the word count command just to see how many lines this bed had. It has, in this particular sample, had 1,032 lines of deletion. So of course, calling this like this would take quite a while. Even if you're doing it in something like a for loop, it's still quite slow. One thing that I like to do is use a command line tool called gargs. And essentially, gargs is essentially just a command line utility that allows us to pipe lines of a text file. So for example, like a bed file, I can pipe in individual lines of that and then use each of those lines as a set of command line arguments for another command. So in this case, Sandplot. And this essentially just allows for easy iteration as well as parallelization of tasks. So essentially what I've done is in the setup script, I've copied the gargs binary to this location, the tilde/bin area. So you can basically just invoke it just by calling it like that. But essentially what's happening here is I'm taking whatever our bed file was, so in this case, our set of deletions, and the cat command is just printing it out into the standard output buffer, essentially. And so then we can ignore this. This is essentially just me changing the genotype from a numeric just to a word. So I'm basically taking, for example, I'm converting 0, 1 and just changing it to the word het for heterozygous. And then I'm changing a 1, 1 homozygous alternate genotype, and I'm just converting it to the word alt. So set is basically just a kind of like a find and replace command in the command line. So we're basically piping this bed file into gargs. And basically what it does is, by default, gargs will parse each line and look for tab separated entries. So for example, the first entry is going to be the chromosome, second entry, or let's say 0th entry is going to be the chromosome, because we start counting at 0. And then the first entry is going to be the start position. Second entry is going to be the end position. So basically by parsing a bed file, I can basically use each element of the bed file as a command line argument, and then basically repeatedly call this plot command, as opposed to having to use some sort of for loop. The second benefit of doing this is that gargs is a tool that allows for very easy parallelization of tasks. So basically by piping these sets of command line arguments into the command that we want to run, I can, for example, do a -p and then set the number of processes. Let's say I have a 32 core machine. I can set this to 32, and it will parallelize this across 32 cores, which, this is basically just a very easy way to achieve parallelization for independent tasks. But even if you're not doing that, if you're just doing the tasks serially, it's just a nice, simple way to take a set of arguments and then just passing it through a command with relatively little effort. All right. Moving on to that. So this is basically just the command that we just showed. But of course, generating 1,000 images still takes a bit of time. It takes, from my testing, it took about maybe 10 to 15 minutes to generate all images of just deletions. So what I've already done is, in the discovery environment, I've basically uploaded the images from the Bcf that I've created here, and put them, in this particular case, just for XH82 in the directory XH82 images. So if you're logged in and at this location in the data store, you can actually just explore this, click on various images, and just view them pretty simply. So for example, something like this. This particular one was a 13 megabase deletion, which from what I'm looking at here doesn't look like it's a true positive at all. It looks like just a region with poorly aligned reads, where there's just like various like spikes of coverage with small like no coverage regions interspersed throughout, essentially. So this is indicative of probably what is a poorly behaved false positive region. But basically all the images are present here, and you can kind of explore that. But I've also kind of extracted a few and put them here as well. So these regions here. So for example, this one looks to be for a pretty surely heterozygous deletion, where you see the normal coverage is about at this region, and then the dip in coverage is fairly apparent, even though it's still a fairly noisy area. But in addition to the loss of coverage here, you're also kind of being reinforced with this notion of, you're seeing a lot of these paired end reads that are aligned at a very large insert size that are spanning the breakpoints of this deletion. So that's basically even more support for the notion that this is essentially a true positive deletion. Whereas, for example, this one is basically about as clean as it gets as a homozygous alternate deletion, where you see a read that essentially spans the breakpoints. You see a pretty much total loss of coverage here, and then you see essentially normal coverage flanking these two regions. But for example, this one seems to have a lot of discordant reads that are spanning the breakpoints at this large insert size, but it seems to have been confounded by the fact that there were these large spikes in coverage in between the purported breakpoints, whereas the midpoint here seems to have like just normal-ish coverage that's a little bit noisy. This is more than likely just a false positive.
So to add some context to this specific image, those spikes are likely repetitive regions, and the alignments that span between them, there's likely some homology between these two regions, and so the aligner just likely incorrectly placed one end of the pair where it was a normal pair in read. One of them just happened to align to a distant, highly similar region. This is a case where we would have liked our depth filter, our exclude regions, to have not called this, but they still definitely pass through, even with our exclude regions, because this is, although there's a huge spike for one reason or another, this was not excluded. If you don't do an exclude region, you're likely to see mostly variants that look like this.
Yeah. And also another reason why this might have passed the filter is because when we computed high coverage regions, we used a very large window size in this particular case, just to make sure that it runs as quickly as possible for demonstration purposes. But if you choose smaller window sizes, you might be able to catch something like this a little bit more easily, because the average coverage is across this genome should be somewhere around here, around like 20 probably, whereas this is going to be more than 4 times the standard deviation above the mean if we had used a smaller window to create our exclude region. So that's something that you can kind of fine tune yourself when you're trying to design your own exclude regions. And that's something you kind of have to determine, perhaps experimenting on a single sample to see how well your exclude regions are excluding regions like this. All right, and then of course another one of these regions, similar to the one we looked at first, where this is like a 1.7 megabase deletion, and it's just lots of ups and downs in coverage, but it just seems like there's just it's a poorly behaved region that confounded the SV caller. All right, so with these visualizations, it's pretty easy just to look at an individual image and see whether they appear to be false positives or true positives. So that kind of leads us to this notion that we can use Sandplot as a manual curation method for large sets of structural variants, and basically using them to be able to accept or reject a structural variant. So one tool that was developed as part of the Sandplot suite of tools is essentially this thing called svplotIt, which is a tool that basically takes Sandplot images and uses Amazon S3 to basically host images in the cloud, and then creates a simple web interface for individuals to be able to kind of go through interactively with various SV images and be able to accept or reject individual SVs. So the idea is you can take your set of VCF regions, you generate Sandplot images from them, and then you can essentially upload them to Amazon S3, and then basically use the web interface for svplotIt to basically have an interactive way of saying, yes, this is a structural variant, or no, this is not. And then you can basically kind of federate this across multiple individuals that can then curate these sets of SVs. So then let's say your lab has multiple people. You can basically send one of these interactive websites to each one of those people, and then they can basically annotate each region and say whether they accept or reject a particular region. And then from there, you can essentially collate all that information and use that as a statistic, use that as like a score for whether you want to keep an SV or reject it as a false positive, essentially. All right, but of course, svplotIt is good for removing false positives from a call set, but in this particular case, we reported close to 1,000 deletions just from one chromosome. So if you did this genome-wide, you'd probably end up with thousands of SVs. So doing this across a large population is still very time-consuming. So it would, of course, be of use to have some sort of automatic curation method to kind of supplement this method as well. So can we somehow filter out these false positives using Sandplot images without having to just manually comb over this data set, which is still relatively slow compared to like more automatic, like computational methods. And that's essentially kind of the motivation for the development of Sandplot ML, which is essentially a machine learning model that we developed and trained using Sandplot images. The hypothesis being that if a human can essentially discriminate between a true positive and a false positive structural variant, then more than likely a machine learning model like an image classifier should be able to do the same thing fairly easy using an automated method, essentially. So the idea here is we're taking Sandplot images, training what is called a convolutional neural network, which is a very commonly used machine learning model for image classification tasks, and using that as a Bcf annotation method, as opposed to just manual curation. All right. So basically, at this point in time, Sandplot ML is essentially provided as a Snakemake pipeline at my GitHub, basically located in my username mchowdh200/Sandplot-ML. And basically, we haven't really explained too much about what Snakemake is, but essentially what Snakemake does is it takes a lot of what we've been doing with individual scripts and individual conda environments, and it basically abstracts a lot of that away. Snakemake is what you would call like a workflow description language, and allows you to basically create discrete steps of a data processing pipeline, for example, SV calling, or running this model with structural variant data, and basically separates them into individual steps. And each individual step can have its own set of dependencies, for example, using conda environments. But now you don't have to manually activate and deactivate your conda environments throughout the process. It'll just basically go through that and do that automatically. So the process for doing this is actually fairly simple. And I've essentially created a way of doing that by just filling out a configuration file. And in this case, I'll just give an example of what that might look like. So a configuration file for the Sandplot ML model is essentially you're using a list of samples, you're providing it with the location of your reference genome, and the location of your reference genome index, as well as location of your Bcf, and then providing with some stuff like the output directory. But then once you do that, running the actual command itself doesn't really require all that many command line flags. You can just run it by just invoking Snakemake. So you just use Snakemake, you provide it with the file that you want to actually, the snake file that you're going to run it on. You can provide it with a number of cores that you want to parallelize it over. In the case of our Cloud Shell, I believe it's only got four cores. So for example, in that context, you would use -J4. But then by essentially saying use conda, you're essentially allowing Snakemake to create all the conda environments and then manage all of your dependencies without having to do it yourself, essentially. So just by having a provided Snakemake pipeline, you can just run this without any extra dependencies besides just Snakemake itself. All right, so again, with generating Sandplot images and running the model over this, I would say it takes about 10 to 15 minutes for Sandplot ML to run over 1,000 images. So what I've done is I've also provided basically the output of what Sandplot ML does after running it in this Cloud Shell environment. So what I've done is in the discovery environment, I have placed what the output of a Sandplot ML run would look like. So going back to the data directory, I have a xh82_image Sandplot ML directory. And basically, it just has the outputs of everything that you would have in there. So for example, the output annotated Bcf would be located in the Sandplot ML results directory, and that would basically contain a Bcf that has various additional format fields added to it. So prior to that, we would just have the genotype and various other SV typer annotations, but now after running the model over it, we provide it with essentially what is an updated genotype, as well as providing the old genotype just for reference to see if something has changed or not. But we also provided with additional format fields for the probability distribution of the model's prediction for each region. So for example, the probability of homozygous reference, probability of heterozygous, as well as the probability of homozygous alternate. So then based off of whatever the highest score there, that's essentially what the prediction would be. So with that, going up here, I've also got this present in my environment already. So if I go to the XH82 Sandplot.ml directory, I can go to wherever the Sandplot.ml results is and look at the BCF there. So of course, like I was saying, looking at a BCF is not conducive to easy reading. So what you can do is just do another Bcf Tools query. And instead of just looking at the genotype and the region, we can annotate it with some additional stuff. For example, we can annotate it with whatever the old genotype was, whatever the current genotype is, the updated genotype from the prediction, as well as the prediction scores that gave rise to this predicted genotype. All right, so if I do that, you can output the prediction bed there and then kind of see what's going on here. So prediction bed. So in this case, we have the region here, the SV type, of course, we know it's a deletion. And then we have old genotype and then new genotype in this case. All right. So if the genotype didn't change, then we're not necessarily all that interested in it, because we're not rejecting, or we're essentially just accepting what the initial call from SV typer was. So we're really interested in looking at calls that went from what is essentially thinking it was a positive SV call into something that goes negative. So going from, say, like 1-1 to 0-0 or 0-1 to 0-0, for example. That means that we've essentially flipped the decision and decided that we found a false positive. In some cases, you'll see some NANs, or not a number, predictions when you see something that was initially already a 0-0, and that's mainly because Sandplot.ml didn't actually provide a prediction for that region because it was already homozygous reference, so we don't even look at it. And in some cases, you will see, like where SV typer fails to provide a call, it'll also provide stuff like not a number predictions as well. That just essentially means that Sandplot.ml didn't even look at those regions.
Hey, Murad.
Yeah.
There's a question in chat about Sandplot.ml working with other, like VCFs from other SV callers. I don't remember. Are there specific requirements for the tags for image generation?
It does not. Basically, as long as the region works to generate in Sandplot.ml, it should be fairly universal. So for example, when we were doing analysis for the initial development, we used both lumpy, smooth essentially, and we also used Manta, which is another SV caller that we haven't really mentioned here, but is another commonly used and effective SV caller. So it does work with others, because essentially all Sandplot.ml needs is just the VCF with, what you would expect is like the normal information, like, the position, the samples, a lot of that stuff very common amongst the various SV callers. We're not using any particular format tags besides something like a genotype or something. So it should work, presumably. Yeah. So yeah, we've got this bed file. So what we can do is we can kind of just examine the results and see, let's look at some of these ones that flipped from thinking it's a positive to a negative, essentially. Of course, we don't have a set of like a gold standard set of regions, so we don't know whether some of these predictions are correct or not, but we can at least examine some of them just to see whether they make sense or not. So the first one I noticed was this should be present in this bed file, the 174013. So that went from a homozygous alternate to a homozygous reference call. And it seems to me that that's a correct assumption. Essentially what's happening here is you have this region of basically no coverage essentially, and even in this region here, it's still below average coverage. So it seems like there's just poor alignment quality in this area, and that low coverage seemed to have confounded the SV caller, whereas the model itself has been trained on images that look like this and has been told explicitly that images that look like this are essentially false positives. Essentially using, in a sense, the same types of intuition that we would use when we're visually validating this. To me, this looks like a false positive. And so as a result, by me providing the type of training data that said that this was a false positive, the model will also make that type of prediction. Let's see, let's look at some other ones. So this one here is going from a homozygous or heterozygous deletion to a homozygous alternate. So it's flipped the decision again. This one is a little bit more difficult for me to make a real call on. This, I could, in a sense, think that this might be a true positive, but it's borderline in my opinion. And it could be a false positive from what I'm seeing, where you just have some overall low coverage. This is, this coverage is still below average, what you would consider normal coverage, and then just like dips slightly below that. And then there's just some discordant reads that are kind of spanning the breakpoints. To me, that's fairly weak evidence. It's probably enough for the SB caller to make a decision here, but in the case of Sandplot ML, it essentially just flips that decision, and in a sense, seems like it's probably the correct decision. And then next, this one here, this one was called as a homozygous alternate deletion, mostly because there was just like no coverage in this region at all. And then the two flanking regions, like right next to the breakpoints, had spikes in coverage, which essentially once again confounded the SV caller into thinking, oh, there's a dip in coverage, it must be a deletion. Whereas if you see like throughout here, there just seems to be overall low coverage here. The coverage is like 4x, 8x here, fairly low. So even though there's like two spikes in coverage here, it's more than likely a false positive. All right. So yeah, are there any questions regarding this?
There was one question in the chat about the sharing the long read model for Sandplot ML. It's not in the GitHub.
Yeah, it's I have to check and see where it is, but we did have a long read model, and basically, long story short, the amount of data that was used to train it was far less, just due to the kind of dearth of long read data that has labeled VCFs, essentially, where we consider like the SVs like a gold standard. So because of that, deep learning methods like convolutional neural networks, these methods are very data hungry when it comes to training. So when you train it on a very small number of regions, it essentially, you're just not going to get the same level of generalization to various SV types as you would with the base model that we had, because essentially, for those that don't know, the base Sandplot ML model was trained using the 1,000 Genomes project set of alignments, which consists of over 2,000 samples, and that essentially equates to tens of thousands, if not hundreds of thousands of SV regions. And so that's quite a bit of data. And then we also had a method of basically determining what we thought was a set of false positives by sampling regions of the genome that were poorly behaved. So we ended up with a very large training set for that. So compare that with the long read model, the performance was really not anything to write home about compared to the performance of our short read model. And that's mainly just because there was just more data available. But something that we kind of have been thinking about for future work is basically using our base Sandplot ML model as almost like a warm start point for training further models, and basically applying what's called transfer learning, where you just take a model that was trained on very large data, and then if you want to fine tune your model on a smaller, more like boutique data set, then you can basically just start off with the pre-trained model and just train with the small data set to kind of fine tune it to your particular application. All right.
Thanks.
Yep. Anything else?
I think that's it from the chat.
All right. So yeah, this is essentially the end of what I have here. Now, there's a little bit more by way of analysis available in the worksheet. For example, looking at like transposable elements and analyzing that stuff as well. But that would be more of stuff like that you would do in the worksheet. But anything else, of course, we weren't able to run a lot of the stuff just due to the nature of how long it would take in a live coding example. But like I was saying, I do essentially have everything that would have been output by this within this data store environment. So under this AG2PI workshop 2021 December, under data, we basically have what would be the data files that we use, all of the output files. Within the GitHub repository, we have the list of all commands used, as well as all the content environments that you would use to set up a Ubuntu or Linux style environment. Besides the bed tools installation, that's literally the only command that we used sudo with. And in theory, you can actually create a content environment with bed tools in it as well. So in theory, you don't really need to use any administrative privileges to be able to run this in a Linux environment. So in theory, you could run any of this stuff in say like a HPC environment where you don't have root privileges, and you can just use conda to basically install these types of tools and run them like that.
All right. I think we should, instead of churning through more of the worksheet, I think we should probably stop right now. Devin did a lot of, as Murad has, Devin did a lot of extra stuff on the worksheet about identifying and extracting mobile elements and characterizing them. So his email is at the top of the worksheet, and the analysis and all of his steps are in the worksheet. So please take a look at that.
Yes.
But yeah, I think
we'll also be. Oh, go ahead.
No, you go ahead.
Well, yeah, I was just going to say we'll also be updating that worksheet to contain some of the Sandplot and Sandplot ML stuff that we have here, and actually we should also be making these slides available as well. I may just put the slides in our GitHub repository for that.
Yeah, maybe we can take the last couple of minutes just to, if anybody wants to chat about something. We are as I said earlier, we are putting together all of this into a nice workflow that would make it very easy. You basically give a list of fastq files from your sample, give a reference genome for your sample, and it would basically do this entire process, including extra transposable element and repeat analysis. We're in the process of developing that. We would like to put together a consortium that's interested in basically non-model organism structural variation. There's a lot of problems that pop up in between species that are kind of common across them, and we'd like to have a group that can share innovations, and so there's not a lot of reinventing the wheel, which in many cases there is in between, when fish biologists don't talk to tree biologists a lot of times, and so there's a lot of the same innovation happening, which is kind of a waste. So if anybody's interested in joining or getting updates about that consortium, it's definitely still in construction phase. But yeah, we'd be happy to chat about how you could possibly be involved or help or just get the products of that group. But otherwise, we're happy to sit here for another few minutes and just talk about structural variation or some of this process.
So I do want to just thank Ryan and Murad and Devin for all the help that they've done today. And just remember that the goal of AG2PI is to build these new communities, specifically crossing organismal boundaries, as well as a variety of scientific domains. And when it comes to, for example, the working group that Ryan is putting together, please keep in mind that the AG2PI does have seed grants that are available in order to help with new initiatives of that kind of type there. But I really want to thank Ryan and Murad and Devin for all of your help today. Just a fantastic workshop. And also, if people are interested, this workshop is recorded and we will be posting it online, so that if you have a collaborator that is interested in reviewing this material again, we'll do everything we can to make it available to you. And with that, I definitely want to give the speakers a big round of applause. And thanks everyone for their attendance today. And Ryan and company, thanks for hanging around online and answering more questions if they're out there.
Yeah, thanks for having us. This was fun. Yeah,
fantastic.
Murad and Devin put a ton of work into this too. So thanks, guys.